Hello everybody, I'm Federico Ponzetto and I'm a postdoc research fellow at the University of Turin. The work I'd like to share with you today was performed at the Department of Medical Sciences of the University of Turin in collaboration with the Swiss Laboratory for Doping Analysis of Lausanne and deals with the monitoring of novel markers of blood steroid profile by a single-run LC-MS analysis. The research on the so-called blood steroid profile has begun quite recently, with the Lausanne research group that in 2016 firstly proposed the longitudinal monitoring of blood concentration of endogenous steroid hormones for the detection of testosterone abuse. This first study highlighted testosterone and DHT as promising biomarkers, in particular for transdermal testosterone administration and for individuals bearing the deletion of UGT2B17 enzyme. In 2019, the IAAF was the first international federation to use such tool for anti-doping purposes, and after the detection of testosterone doping in two female athletes, WADA officially stated that blood measurements could constitute a further tool for anti-doping organizations. Therefore, the research on this topic moved forward to the discovery of additional blood markers of testosterone doping, and thanks to the metabolomics approach, the improved sensitivity of androgenous phase II metabolites was observed and reported. Last year, the group of the Stockholm WADA-accredited laboratory developed a reliable LC-HRMS method for the simultaneous measurement of a large panel of androgenous phase II metabolites and applied it to a small population with satisfactory results. Finally, just some weeks ago, a novel study has been published comparing the performances of blood and urine markers analyzing for the first time samples of a female population administered with testosterone gel. This last study proved once again the complementarity of the urinary steroidal module of athlete biological passport and the recently proposed blood steroid profile. The aim of the present study was to develop and apply a first analytical method capable of measuring in one single analysis all reported blood markers of doping with endogenous anabolic androgenic steroids. The target analytes of the LC-MS method we developed are endogenous steroid hormones. As you know, this class of compounds includes a high number of molecules that are all synthesized in human organisms starting from cholesterol. In our method, we decided to include not only testosterone and DHT, but up to 13 free steroids, which are routinely measured for clinical purposes and could be informative of athletes' health conditions. Furthermore, we know that all endogenous steroids are subject to both phase I and phase II metabolism. And as explained in the introduction, we'd like to monitor with our method all relevant doping markers belonging to the class of androgenous phase II metabolites. In particular, we decided to monitor 9 glucuronides, including 5 different isomers of 5-androstenediol glucuronide and 8 sulfates, in order to cover all the recently reported markers of testosterone doping. Because of the large presence of isomeric compounds, the main challenge during method development was represented by the chromatographic separation. The latter was achieved after testing the performances of various LC columns with different chemistry by using a core shell column, the Kinetex PS-C18 in its 15 cm format, and by using water and methanol with ammonium acetate buffer as mobile phases. With these conditions, we were able to efficiently separate all target analytes and in particular all isobaric isomers which are depicted herein with the same color. In the final method, the sample preparation procedure has an utmost importance. Indeed, sample cleanup protocol should be capable at the same time of extracting all target analytes, which own strong differences in terms of polarity, and reducing matrix effects as much as possible. For this aim, we chose a solid phase extraction using the 96 well plate format. The extraction protocol starts from 500 microliters of serum or plasma, and it is capable of extracting all target steroids with a recovery higher than 80% coupled to negligible matrix effect. The whole procedure takes 2 hours to process 48 samples and could also be partially automated. For the instrumental analysis, we adopted the chromatographic condition displayed before, with a flow rate of 300 microliters per minute, keeping the column at 55 degrees and injecting 20 microliters of the sample. MS analysis was performed in polarity switching mode, and for each steroid, 2 MS transitions were selected and optimized. Thanks to the developed method, we are able to quantify the 30 target steroids in a single run analysis of 25 minutes. The method was then used to perform a preliminary study analyzing real plasma samples collected at the blood bank of the Turin University Hospital. The obtained results, expressed as mean plasma concentrations, are presented in this table, and method's quantitative performances were checked by injected in each batch of analysis quality control samples, part of a commercial LC-MS kit by Chrome system, which gave satisfactory accuracy outcomes. The concentration measured in this study showed good coherence with reference intervals already reported in literature, most of them referring to free steroids, and highlighted novel information regarding both the circulation of 5-androstenediol glucuronides, with the 17 glucuroconjugated forms being the most concentrated, and the impossibility to detect some androgenous metabolites, such as DHT sulfate. Finally, the high concentrations measured for some doping markers, such as epiandrosterone sulfate, confirmed the possibility of developing RMS analysis in blood, as recently proposed at Manfred Donicke workshop. The next step of this project will now be the transfer of the method to the Lausanne WADA accredited lab, followed by the investigation of reference values of all proposed markers in a wider population, and by the evaluation of the effects on blood steroid profile of possible confounding factors, such as circadian rhythm and physical exercise, for which dedicated clinical studies have already been launched. An additional interesting perspective will also be represented by the assessment of blood micromatrices for this kind of measurements. In conclusion, I'd like to thank all the collaborators that work at this project in Turin and Lausanne, and the PCC Fellowship Program for supporting me and this research. Thank you.